Hi, how are you? You already started the room. Perfect.
Yeah, I'm good. Thank you. How are you,
Thank you.
Katrina?
I'm good, good. Thank you. Could you do me a favor and
Sure.
make me a moderator so I can help you with a lot of different tasks?
How can I do it?
So you click on my profile picture,
Mm-hmm.
and then on the bottom should
Okay.
show view full pro. Ah, perfect.
That
Thank
should
you
be
so
done
much.
now. Yep.
How's your
Perfect.
weekend going? Everything good?
Not too bad. I have a conference next week in Scotland, so I was preparing for it.
Ah, okay.
Yeah.
That's nice in Scotland.
Yeah, after a long time, this is going to be like in-person conference, so pretty excited about it.
Yeah. That's wonderful. Where
Yeah.
in Scotland?
In Glasgow is
Okay.
the IBNS conference.
I'm happy for you. I hope
How,
you'll have a
how
lot of
was,
fun.
thank you. How was your weekend?
Good, good. We traveled a little bit. Friend of ours, they had their daughter's birthday party, so we went to Long Island. It's not too far,
Nice.
but
That's nice.
yeah, yeah, we are friends for what the trying to bring um, him actually to Clubhouse, but he works at Brookhaven at the Particle Collider.
Mm-hmm.
But his work is mostly DoD, so he always feels like he cannot tell like, what he's actually doing, so that wouldn't be too interesting.
Right.
So,
Yeah, you already have like a lot of members here in Clubhouse. I mean, I was listening to some of the talks, and I mean, you do cover like a broad, like aspects of like research fields. So that's amazing.
oh yeah, thank you so much.
Yeah.
Yeah, I have, we have a lot of different interests here, members from all kinds of backgrounds, so yeah, that's how it comes together to have such a variety of talks. So, hi,
Right. And
Jamie. Yeah, sorry. Go ahead.
yeah, and then you have somebody might have said something.
Oh, I just said hi.
Hi.
Really looking forward to your talk. I
Yeah, I'm
probably
very
gonna ask
excited.
you. I'll probably ask you an astrocyte question toward the end.
Astrocyte question. Okay. Sure. Thank you.
Hello, Doctor. Thank you very much for your time, and I'm looking forward to your talk as well.
Thank you. Thanks, Jimmy. Looking forward to the talk. I mean, I'm excited to be in this platform today. I
And
mean,
I'm also in Scotland, so I'm glad that you're having conferences and stuff here. That's great.
oh, you are in Scotland?
Yeah, I'm in Edinburgh.
Edinburgh. Okay.
Yeah,
I'll be
yeah.
passing through Edinburgh. I'll be going to Newcastle for like meetings and a presentation also. So I didn't find any like direct train from Glasgow, so I'll stop over in Edinburgh for maybe like half an hour.
Ah, well, I hope you enjoy it.
Thank you. And this is one of the best thing in this platform that I'm seeing, that like people from all around the world can actually participate and share their like knowledge and like advancements in research. So that's very exciting.
It's definitely been one of my favorite things about this. People from all over the world have
Hmm.
been speaking here, and I've been so lucky to be part of that. So you're very much right, and Katerina has arranged an excellent group, and a lot of interdisciplinary specializations have been discussed here. It's been really quite exciting, and I'm just really pleased you're here to offer your findings with us as well. This is just very exciting.
Yeah, I mean, Katerina is doing amazing work here, and there are like talks, like regular talks, almost sometimes like 5 days or 6 days a week. So like organizing with all the speakers and then like diversifying the like topics of presentation. It's a lot of work, so you all are doing amazing work there.
She really has done an amazing job, and just the breadth and depth of the speakers that come by has just been a a nightly experience for many of us.
Mm-hmm. Yeah, absolutely, totally agree with that.
Yeah.
Thank you so much. Please don't make it about me. It's so wonderful that so many people actually agree to come. It always surprises me because there's, well, we try to. So the thing why I set up Spotify and YouTube account is actually, I don't advertise it. I should, but it's more that the altmetric score of the papers go higher.
Mm-hmm.
So, but other than that, it's purely altruistic, I think, for speakers to come here and share their time with us. So, thing that people do that because people don't get paid, or there's no direct outcome. So,
Yeah, these
I
are these
think.
are all like voluntary participation,
Mm-hmm.
but like the driving force is the passion for science, right?
Yeah, exactly.
Yeah.
So when COVID hit, it annoyed me very much how bad image of people were on social media,
Mm-hmm.
also on this platform and on other platforms. So yeah, I tend to try to do something instead of just sitting there. And then people are actually very interested when they feel like people actually are genuinely interested in what they're doing. They also become interested. So yeah.
Right. Yeah, absolutely. Because I mean, here, like we are talking in front of like a broad audience, which like brings more like chances of like collaboration. Like more people will know about your work, like whoever is presenting, and I mean that, and then the talks are all recorded, so like it can be like viewed later. So I think this is all great.
I can personally speak to this as like an education for me.
Hmm.
I've came to the clubhouse and Katerina's club and science society, not knowing half of this stuff just simply a couple of months ago. So this could be in some kind of a science society academy down the
Hmm.
line. All the past talks, it covers so many different topics, so many different areas. Um, but after a while, like you said, there's been so many willing speakers that just for the chance to talk about what's been passionate to them, and so many people willing to listen. I've actually managed to find a lot of crossover. Finding about neural plasticity and finding things about physics and things about chemistry that I never ever had a clue, but they keep coming up. There's common threads, which is quite exciting to hear a speaker talking about one thing and another speaker talking about something that's got a side-on aspect to it. It's been even happened, hasn't it, Katerina and Serena, when we've been able to say to a speaker, "Oh, do you know of so and so's work?" Because we just had them as a speaker,
Hmm.
and perhaps they could collaborate. It's been quite exciting. It really has.
Yeah, that I mean I totally agree, and that's what I also strongly believe. Yeah.
Well, it really is. There's just so many advances happening,
Hmm.
and the multidisciplinary nature is really notable, and just how it seems. I've always had an interest, but it really seems like we're in a period where the advances have just accelerated so much. It's just incredible.
Oh yeah, with like technological advancements and like a lot of different things, it's like every week. I mean, I'm talking about like my own field in like transcription and
Hmm.
gene expression, epigenetics. Like there are like new techniques coming up like every now and then, and it's amazing to like it's so hard also to keep up with all the advancements. So how they can be used in biology or like other fields, and then what's the actual application, and for those you need to like listen to like people who are actually doing it. And sometimes, well, most of the times papers like publication takes time to happen. So basically like what is there in the paper is the work which was done like for the maybe like past 5 years. So I mean, I'll present in my work today. In my presentation, I'll mostly talk about published things, but I can also talk about some of the new advancements, also like technological advancements. And I did notice
Oh,
in like
good.
several other talks here in this platform that like real cool techniques and the applications. It's so fascinating.
Welcome, Doctor. How should I pronounce the snake?
You can call me Jyoti. My pronunciation is Neha Jyoti, but people call me
Jyoti.
Jyoti. People call me
Jyoti.
Jyoti. Jyoti, yeah.
Yeah,
Jyoti is
Jyoti.
fine.
Yeah. We really appreciate your
Thank you.
come to our platform and the very, very positive feedbacks. And so it's actually also I second that it's a very multidisciplinary and under the great initiative from Katerina. I still don't know how she can do it. I mean that's a lot of work inviting so many uh, excellent researchers from across all the field. So actually, I'm curious. Maybe in one of the future sessions we can invite some of them back and from different field and
Mm-hmm.
have them, for example, Jody and from his perspective that how this general public like platform like Clubhouse can help. I do see a strong interest from some of the researchers that they without the Clubhouse, they're already very actively connecting, seeking the public audience, right? So, uh, so this general public audience is one, and there are also colleagues, professional researchers on Clubhouse as well.
Mm-hmm.
So I myself, being a research engineer, not in the academia but in the research industry, but educated in in similar programs, research programs. So so that's I think this Clubhouse being one platform. There's others. This is really itself is an experiment. I think that's probably Karina. I haven't discussed this with Karina and Serena and Jamie
Mm-hmm.
and others yet. But I think this is itself. I think is worthy research itself. I think how because as researchers, we often specialization and being general and being specialized, right? So
Right.
it's a trade-off, but often specialized. That's what is required. Then what is the most productive way of reaching out, right? Connecting to the application and even colleagues in the same subfield, not being constrained physically by physical space, right? That's what I think the COVID uh, pushed for new innovation like Clubhouse, which I think it will stay. Yeah, I mean it's this is essentially the most strict down version of it. Just all it takes is voice communication, right? So of our thoughts.
Yeah, yeah. That's true. Yeah.
Okay, shall we start? I think we can slowly start our session.
Sure.
So, yeah, thank you so much, everyone, for coming and welcome to the Science Society. We have our we are honored to have our guest speaker here today, Dr. Jyoti Chatterjee. I hope I'm saying it right.
Yeah, that's correct.
Thank you, sorry. And let me give you a little bit of information about our guest speaker. He did his bachelor in science and microbiology at the Surendranath College, Kolkata, and his master's degree also in molecular biology and genetics at the Presidency College, Kolkata, and his PhD in neuroscience from the, oh, that's French, Laboratoire d'Imagerie at neuroscience cognitive neuroscience at the University of Strasbourg in France. I've been in Strasbourg a lot of times because my mother used to work for the European Parliament. So
Oh, okay.
yeah, growing up, I was. It's a beautiful city,
It is.
but
It is a beautiful place. Yeah.
yeah, but let's. And he's currently working at assistant research scientist in the laboratory of Professor Ted Abel at the Department of Neuroscience and Pharmacology at the Iowa Neuroscience Institute, the University of Iowa. And yeah, we are very honored to have you here. It's an amazing research in memory. And congratulations, first of all, to your wonderful paper and this really interesting work and very important work. So, if it's okay, Jamie would ask you a couple of general questions first, and
Sure.
then the stage is yours. Thank you.
Yeah. Sure.
Thank you very much, Katerina. Okay, so first of all, Doctor, thank you very, very much for joining us. This is actually quite exciting. One of the things we actually do is we ask a couple of questions about what got you started. So I'll begin with saying, what was it that first got you into science itself, that started you on this path of academia, as it were? Was it something in childhood, or did you start later on? Anything you can tell us, we love everything here.
I love this question. uh, I mean, I was always very like passionate for towards like innovation. Of course, when I was a kid, I had no idea about like what like researchers or scientists do, but I can recall some like some things I used to do when I was a kid is like I used to extract it. It may sound crazy, but I used to like make extracts from like plants, different plants and flowers in India, and I used to be like obsessed with the different colors of extracts I obtain, and I used to believe that these are all medicines. So I mean, yeah, I wanted to like develop medicines from like plants, and uh, but then when I started my like after masters, I joined a lab. That was basically the first experiments or things that I did, that I collaborated with some chemists and asked them to extract the molecules obtained from some of the plants that we often see in India. And that actually laid the foundation for my PhD work, because one of the drug that I found was the first activator of a histone acetyltransferase. And then, I mean, rest is like history to me, because I mean, there was this drug showed to be like enhancing memory, and then showed potential for like memory rescue in Alzheimer's. So the dream that I had in my childhood, I could pursue it when I was doing my PhD. And yeah, I think that's what like drove me. And I was always interested in science. Biology was my favorite subject, and I think about like innovations, coming up with new things. Yeah, that's it.
That's absolutely fantastic and wonderful to hear. Did you find that when you studied, even in childhood, when you were pulling the plants apart to see how they ticked, did you find that the more you discovered, the more there was yet to discover, and the more fascinated that you got?
I had no idea what I used to do in childhood days. I had no idea, but yeah, I mean, coming up with like new ideas for like doing something. Most of the things like ended up being frustrating, because like I'll extract something today, and tomorrow when I wake up, the color like turns to more pale colors. I used to be sad. Yeah, but I mean, I used to try it the next day. Again, it may sound like crazy.
Oh no, no at all. It sounds amazing, like your curiosity was driving you. And what then led you there to, well, you were starting to actually talk about that, and brought you into discussing into memory and everything, and where you got to here. Just how do you get to this point where we're at right now with the paper?
I'm sorry, could you please repeat the
Beg your
question?
pardon. What was it then that got you focused on this specific aspect of long-term memory and led to the paper that you're going to discuss today?
Yeah, so. I mean, again, that drug that I found, I started my career in cancer biology, and I was like screening different drugs for like potential role in cancer treatment. And the drug that I just mentioned, was like showed to it was found to be activating a particular enzyme that enhanced memory. In my hand, it enhanced memory. So that's where I shifted my research to like neuroscience, because I could see that it should enhance memory, but nobody have ever like tried it. And I had like no experience with like behavior. I went to France for 3 months to try these experiments, and I mean, doing those behaviors and then learning like how animals could react to a certain environment, and then how can like memories be formed, and like understanding this memory mechanism, especially like worldwide. Dementia is a big threat, and like with elderly populations, like there is a big impact in the like society with dementia and age associated memory decline. And then if we come up with some I mean, again, NIH is also like supporting a huge amount of research these days, but uh, um, I mean, I could see that it's very important. And my grandfather, he had Parkinsonism, and he had this dementia also. So I have seen how someone who like starts losing their memories, how painful it could be for the person and also for the families. So a combination of a lot of things actually contributed towards my like interest in memory. I'll talk about it in my presentation, like how I came up with this paper. It started from during my PhD work when I was interested in CBP, and then progressively I like went down into the mechanism and like developed the story for or published the paper, which came up more recently.
Thank you very much, Doctor. That's wonderful that you shared that with us, and we would like to thank you. And when you're ready, I think we're all eager, so the floor is yours. Thank you.
Thank you, Jamie, and I'd like to thank Katrina and like all the members here for like having me here. I'm very excited for this talk. So you all have access to the slides, right? Uh, I mean, in this presentation, I'll give some background on from the. I mean, I'll not just talk about the paper which came out in Science Advances, but I'll also provide some background of the work. So throughout my academic career, I have like mostly focused on understanding the molecular mechanisms of memory consolidation, and use those information to develop therapeutic strategies to treat memory impairment in Alzheimer's disease and related dementia models. Slide number 2. Memory consolidation starts with encoding of the information, which is construction of a single memory trace from all the sensory information collected. But to convert that like single memory trace to a stable long-term memory, it requires a post-experience process called consolidation. During this consolidation, the initially labile memory trace progressively or gradually gets stabilized to form long-term memory. And this process requires transcription and translation. Once the memory is formed and once the memory is consolidated, it can be recalled later. So in our lab, we used a spatial object recognition task or SOR task to assess long-term memory in mice. Typically in this task, we exposed an animal in an open field with 3 different objects, all the objects in specific spatial locations. And the animal will explore the objects to encode the information, especially the spatial information of those objects. Then it will be returned back to its home cage where consolidation will occur. And this consolidation requires precisely timed transcriptional events. And then to assess long-term memory, we return back this animal to the open field 24 hours later. But here we move one of the objects to a novel spatial location. If the animal acquired the memory of the object, it will explore the moved object more compared to the 2 other objects in the familiar location. Next slide. This form of memory is dependent on hippocampus. It's called spatial memory, and in humans, hippocampus is found deep inside the temporal lobe, where it controls not only spatial navigation memory but also episodic memories. Hippocampus, it enables construction of cognitive map in the environment around the subject. And the most important or relevance of hippocampus dependent memory in our research is that it is regulated by epigenetic mechanisms through gene expression and is mediated by histone acetylation. And importantly, in Alzheimer's disease and related dementia, hippocampus is one of those sites which shows early pathological signature of the disease, and then it gradually spreads to other brain regions. Alzheimer's disease is also associated with impairment in spatial navigation memory as well as episodic memories. Next slide. So we know that transcription is essential for memory consolidation, and decades of research have showed that after a learning experience, there is a huge induction of gene expression in the hippocampus. Here in a classical experiment performed by John Buzowski, Carol Burns, they showed that in hippocampal subregion CA1, after a learning experience, shows induced expression of activity-induced gene ARC mRNA. And researchers have found that this induction in gene expression or these activity-dependent genes are essential for memory consolidation. But how these genes are regulated? Next slide, slide number 5. So identify the mechanism by which this activity-induced gene expression happens. Researchers identified a transcription factor, CREB. CREB binds to specific genomic regions known as CREB response elements or CRE elements. This CREB gets phosphorylated, post-translationally modified, and activates gene expression. But soon it was realized that phosphorylation of CREB is not sufficient to activate its downstream gene expression. CBP, also known as a CREB binding protein, CBP binds to CREB through CBP's Kix domain. And that binding of CBP to CREB triggers downstream gene expression. Now, CBP is also, CBP, here I'm talking about its function as a transcriptional coactivator, but CBP also functions as a histone acetyltransferase enzyme. So in addition to bind to CREB, it also acetylates nearby histones, thereby opening up the chromatin and then triggering gene expression. And this realization really launched like a lot of recent work in memory and epigenetics. Next slide, slide number 6. So at the baseline level or unstimulated neurons, the chromatin or chromatin around the activity-induced gene promoters are tightly like closed, and that signals for repressed transcription. But following learning or neuronal stimulation, CBP binds to CREB on the promoters of those genes, which have a lot of several CRE elements, and then CBP also acetylates histones, thereby opening up the chromatin and then recruiting the basal transcriptional machinery to drive gene expression. Next slide. So my own work as well as others have showed that CBP interaction with CREB through CBP's Kix domain is essential for memory consolidation. Here on the left side of the slide, we can see that in a mouse model which harbors a mutant CBP Kix domain, which impairs CBP's ability to bind to CREB, these mice shows long-term memory impairment. And then activation of CBP in wild-type mice using a pharmacological strategy, using a nanobiotechnology approach to activate CBP leads to enhancement in histone acetylation as well as enhancement in long-term memory. Therefore, CBP CREB acts as a molecular switch where modulating its function can lead to bidirectional changes in memory consolidation. Next slide. So far we established that CBP CREB interaction is essential for long-term memory consolidation. But how? How CBP, or what are the downstream targets of this CREB CBP pathway? So I first performed a total RNA sequencing from wild-type mice, and animals were either trained in fear conditioning or Morris water maze. Both the task requires hippocampus. And on the left side, we see the genes that are induced after learning in wild-type mice. And we see several immediate early genes or activity-dependent genes are induced in the dorsal hippocampus 1 hour after the training session. But in the CBP-KIX domain mutant animals, there is a huge downregulation of several immediate early genes. Especially, we find these NR4A1 and NR4A2 proteins to be consistently downregulated in different mouse models of mutant CBP. Next slide. So NR4A family of transcription factors or subfamily of transcription factors are the direct targets of Krebs CBP pathway. They belong to the largest superfamily of transcription factors in metazoans known as nuclear hormone receptors. And all the three members of NR4A subfamily, NR4A1, NR4A2, and NR4A3, are induced after learning in the dorsal hippocampus. On the right side, we can see that following a spatial object recognition task, there is a significant induction of NR4A1 as well as NR4A2 gene expression within 1 hour after the training session. And then the expression level goes down to baseline from 2 or 3 hours after the training. Next slide. So how what is NR4A? What are the different like domains of NR4A and how it functions as a transcription factor? NR4A family of transcription factors bind to the DNA through on specific genomic regions known as NBRE regions or the nerve response elements. They bind as a homodimer, but they can also bind to DNA as heterodimers. Therefore, like for example, NR4A1 can bind to NR4A2 and regulate expression of their downstream genes. But these NR4A family members can also interact with other nuclear hormone receptor family members and can also regulate their gene expression. For example, here we can see that NR4A family members can interact with RXR, get recruited on the DR5 elements, and then regulate their gene expression. Now, NR4A are induced after learning. Is it essential for memory consolidation? Marcel Wood's lab showed that siRNA-mediated knockdown of NR4A1 or NR4A2 impairs long-term memory consolidation in wild-type mice. So indeed, NR4A members are essential for memory consolidation. Is it a target for memory enhancement? Next slide. So I used a chemical or a pharmacological approach to target this transactivation function of NR4A proteins. Here I collaborated with a chemist, Stefan Seif, and he developed several molecules or synthetic elements, synthetic agents which can bind to NR4A transcription activation domain and enhance its transcription function. So I'm showing an example for NR4A2 agonist. We have also characterized NR4A1 agonist and found similar results. So this drug, CDM12, it binds to the NR4A transactivation domain and enhances its function. I tested this drug in wild-type mice following a weak learning protocol where control mice will not consolidate any memory. They will not show memory consolidation. But interestingly, I find that treatment with this drug enhances long-term memory in wild-type mice. Now, we know that memory impairment is associated with or age-associated memory decline is a serious threat, and there are not many therapeutic options available. So some researchers have showed that NR4A activation is impaired in aging. And so I hypothesized what happens if we activate NR4A in aged animals? Can we rescue memory? And I administered this drug for 5 consecutive days in aged animals, 20 months old aged animals, and trained them in this spatial object recognition task and tested their long-term memory. The vehicle administered animals showed long-term memory impairment, but interestingly, activation of NR4A2 rescued long-term memory deficits in this aged model, and it reversed memory deficits. So clearly, these NR4A proteins are therapeutic targets, could be therapeutic targets for memory rescue in age-associated memory decline. Next slide. So NR4A family members are important for memory consolidation. and they are potential target for memory enhancement and age-associated memory decline. But how NR4A regulates memory? Now, they are transcription factors, and we believe that NR4A regulates memory through their downstream targets. So, as I mentioned before, NR4A proteins interacts with each other, form heterodimers, and that formation of that heterodimer complex recruits the basal transcription machinery through NR4A transactivation domain to drive gene expression. To demonstrate the molecular mechanism by which NR4A regulates memory, we used a NR4A dominant negative mouse model, where we deleted the transactivation domain of NR4A. So basically here what will happen is the dominant negative NR4A will get recruited to the DNA, will interact or form heterodimers with other NR4A members, but as it lacks the transactivation domain, it will fail to recruit the basal transcription machinery and it will fail to drive gene expression. So gene expression from those locus will be blocked. Next slide, slide number 13. So we first assessed long-term memory in this model and found that control mice shows significant memory consolidation. They show significant discrimination towards the moved object, while the NR4A dominant negative mice shows impaired long-term memory consolidation. But we don't see any memory impairment in short-term memory, where both control and NR4A dominant negative mice shows intact short-term memory. Now, it's important to note here that long-term memory requires transcription and translation, where short-term memory is independent of transcriptional events. So, to identify the downstream targets of NR4A proteins, we next performed a total RNA sequencing after learning. We waited 2 hours after training so that it gives sufficient time for NR4A proteins to express and get recruited on its target gene promoters. We found that there are like 40 genes which were downregulated after learning in NR4A dominant negative mice, and only 14 genes were upregulated. A pathway analysis revealed that several pathways related to the endoplasmic reticulum protein folding machineries are downregulated in this mouse model after learning. Especially we see this misfolded protein binding, chaperone binding, and protein processing in ER pathways. Next slide. I also performed a protein-protein interaction analysis and found that a multi-protein ER chaperone complex consisting of HSPA5, PDIA6, and several other members to be significantly downregulated in this model. This multi-protein ER chaperone complex is known to bind newly synthesized polypeptide and facilitate their folding. But before moving into that aspect of like protein folding machinery, we made sure that, or we confirmed whether this regulation of genes encoding ER chaperones are directly regulated by NR4A factors. We used publicly available database on NR4A1 ChIP-seq and found that indeed HSPA5 and PDIA6 promoters shows enrichment of NR4A1 on their gene promoters. I also validated the expression of these genes or NR4S target genes at the mRNA level as well as at the protein level from synaptosomes. And I always found that this regulation is only seen after learning, where home cage animals which were not trained in any behavior, they don't show this regulation. They show equal levels of expression of these HSPA5 and PDIA6 genes, suggesting that there might be an activity-induced expression of these ER chaperone genes, which is regulated by NR4A. Next slide. So to understand or to confirm whether this expression of ER chaperone genes are also induced by activity, we performed a total RNA sequencing from control or wild-type mice. Wild-type mice were either trained in SOR or just handled only at their home cage. And then the data was compared with the NR4A dominant negative bulk RNA sequencing after learning. And to simplify this graph on our left side, the genes that are labeled here are the ones which shows induced expression after learning in wild-type mice, but are downregulated in NR4A dominant negative mice after learning. And several members of this ER chaperone multiprotein complex were seen to be in this particular cluster. I also verified the expression of these HSPA5 and other ER chaperone genes after learning in wild-type mice. And indeed, their expression level goes up after learning within 1 hour after training, and then progressively it goes down at 4 hours time point after training, suggesting that they are activity-induced genes. And all these experiments were performed from dorsal hippocampus, and we know that dorsal hippocampus is a complex structure. It consists of multiple different subregions, CA1, CA3, denta gyrus, and several others, and also is composed of various different types of cells, like excitatory neurons, interneurons, oligodendrocytes, astrocytes, and so on. So, with the recent advancements in single-cell RNA sequencing, uh, we could actually investigate the expression profile of these genes within individual population of cells from a given hippocampal subregion. Here I'm showing the single-cell RNA sequencing results from CA1, where we could clearly see that CA1 shows induced expression of HSPF5 and PDIS6. I'm also working on the other cell types currently. And to compare this single cell RNA sequencing results, I have also generated some data on single cell ataxic as well as spatial transcriptomics to really understand the molecular signature or transcriptomic signature of these genes after learning. Next slide. So we identified a multi-protein ER chaperone complex which binds to newly synthesized polypeptides after learning, and this complex is regulated by NR4A proteins. Now, we know that learning induces gene expression. A huge amount of genes are transcribed and translated, but we have often ignored the fact that all the proteins that are synthesized after learning, they need to be correctly folded, properly folded, and then trafficked to be functionally active. And we believe that we hypothesize that this ER chaperone complex performs this function of folding newly synthesized polypeptides after learning and then facilitates their folding, their trafficking, I'm sorry. So to prove this concept or this hypothesis, we need a target of this ER chaperone complex. And by literature survey, we found that gluN2A, which is an NMDA receptor subunit, interacts with HSPF5. Now, these NMDA receptors are predominantly synaptic, essential for synaptic plasticity and memory consolidation. Especially NMDA receptor gluN2A expression induces after learning in the hippocampus, and that pharmacological enhancement of gluN2A improves memory. So it fits all the criteria to test its function or test its folding ability after learning. So next slide. We first used a cultured neuron model where we induced neuronal activity or neuronal depolarization using KCL and stimulated the neurons. And we found that activity induces surface localization of gluN2A, whereas NR4A dominant negative expressing neurons fails to show this activity induced surface deposition of gluN2A. We also co-localized gluN2A with PhD95 and found that neuronal stimulation induces not only surface localization of gluN2A, but also PhD95 or postsynaptic density compartment localization of gluN2A, which was impaired in NR4A dominant negative expressing neurons. But most interestingly, when we overexpress HSPA5 in NR4A dominant negative expressing neurons, we could see increased postsynaptic density co-localization of gluN2A, which suggests that HSPA5 is downstream to NR4A, where it plays active role in folding gluN2A and trafficking it onto the postsynaptic density compartments. Next slide. Now, to prove this in animal model, I overexpressed HSPA5 in the dorsal hippocampus of NR4A dominant negative mice. The EGFP-infused animals showed impaired long-term memory, but the HSPA5 expressing or mice that were infused with HSPA5 in the dorsal hippocampus shows enhanced long-term memory, which also suggests that or confirms our findings from the cultured neuron model that indeed HSPA5 is downstream to NR4A and plays active role in memory consolidation. Now, to understand the protein folding aspect of memory consolidation, we used a pharmacological approach. We used or administered a drug known as phenylbutyrate. Phenylbutyrate is a chemical chaperone, a molecular chaperone. It binds to unfolded proteins and facilitates their folding. But PBA is also an HDAC inhibitor, where it enhances histone acetylation. So we used another control or another control drug, sodium butyrate, which is structurally very similar to PBA. It is also an HDAC inhibitor, but it lacks chemical chaperone function. And interestingly, we found that, of course, vehicle infused or administered an NR4ADN mice shows long-term memory impairment, but mice that were administered with phenylbutyrate, which is a chemical chaperone, showed long-term memory consolidation, or they showed a reversal of memory deficits, whereas sodium butyrate, which lacks chemical chaperone function, failed to enhance or rescue long-term memory deficits in these NR4A dominant negative mice. We also found that PBA cannot enhance memory or further in wild-type mice. So taken together, these results suggest that ER chaperone function is essential for NR4A-mediated long-term memory consolidation. Next slide. Now we established a molecular mechanism by which protein folding machineries are essential for long-term memory consolidation, and we used a mouse model that shows long-term memory impairment. And memory impairment or memory loss is associated with aging and dementia, which is a syndrome in which there is a deterioration of memory, thinking, and behavior, and also the ability to perform everyday activities. Like worldwide, 50 million people suffers from dementia, and that number increases by at least 10 million per year. Where Alzheimer's disease itself contributes towards 60 to 80% of cases. Next slide. The two major hallmarks of Alzheimer's disease are neurofibrillary tangles and amyloid beta plaques. For this work, I'll focus on the neurofibrillary tangles because I used two different mouse models of this tau. So neurofibrillary tangles are formed from hyperphosphorylation of tau proteins. Tau in wild-type mice, they are microtubule stabilizing proteins. They bind to microtubule and stabilizes its structure. But upon hyperphosphorylation, it disintegrates from microtubules and starts forming like clump-like structure, which ultimately leads to these neurofibrillary tangles, which are potential biomarkers or potential pathological signature of Alzheimer's disease and related dementia. Next slide. So I first used a mouse model of mutant tau. This mutant tau model harbors P301S mutation, which is often seen in patients from frontotemporal dementia. And we found that this mice shows downregulation of CBP. CBP interacts with CREB and regulates NR4A gene expression. And that CBP itself is downregulated in this model. And there is also a downregulation or reduced expression of histone H2B acetylation. Histone acetylation drives gene expression. So I used a drug, the same drug, CSPTTK21, which is an activator of CBP. Previously, I found that it enhances CBP's ability to acetylate histones, induces gene expression, and enhances memory in wild-type mice. So here in this work, I administered this drug in this tau model and found that activation of CBP enhances histone H2B acetylation. Now, this mouse model shows long-term memory impairment in spatial navigation task, but activation of CBP reverse long-term memory deficits, which is interesting because here what we are doing is we are activating the remaining amount of CBP to make it more transcriptionally active and activates its downstream pathways. And what are the downstream targets of this particular protein? These are the NR4A factors, the activity-induced gene expression, like ARC, NR4A, AGR1, and we performed a total RNA sequencing, a whole transcriptomic analysis to identify all the genes that are altered after learning in the tau mice and the ones which are rescued by upon activation of CBP. And to I found NR4A1 as well as NR4A2 genes were induced after learning in wild-type mice, but they were downregulated in the tau mice after learning, but activation of CBP reversed this downregulation. It rescued the expression of NR4A1 and NR4A2, which also suggests that this pathway is a potential therapeutic target for memory rescue in Alzheimer's disease and related dementia, and that NR4A factors are also important for or might have potential therapeutic ability to enhance memory. Next slide, slide number 23. So I next investigated the expression of NR4A genes in patient samples. I used this Allen Institute's Aging, Dementia, and TBI study, full transcriptomic profile from hippocampus obtained from postmortem patients with Alzheimer's disease and related dementia, and found that NR4A1 and NR4A2 genes were progressively downregulated across increasing pathological stages as measured by SERAT score and BRAC stages, whereas NR4A3 didn't show any significant downregulation. Next slide. So to investigate the potential of NR4A or targeting NR4A in Alzheimer's, we used another mouse model of mutant tau. Now this mice harbors P301L mutation, but it is regulatable. So it is under the mutant transgene is under the TATO promoter, so that we can control the expression using CAM kinase 2 driven TTA transgene, so that the transgene expresses only in excitatory neurons, and as it has TATO promoter, we can regulate the expression of the transgene using doxycycline. So on the Western blot on our left side, we can see that in the tau mice, there is an increased expression of pathological tau or phospho-tau using this Western blot approach, whereas doxycycline treatment completely abolishes the expression of mutant tau proteins. These animals shows long-term memory deficits, but doxycycline treated animals shows long-term memory consolidation. Next, we investigated the expression of NR4A genes in these mutant tau mice. On the left side, we see that following learning, there is a significant downregulation of NR4A1 and NR4A2 in the dorsal hippocampus of tau mice, but NR4A3 didn't change. And this is very similar to what we found from the patient sample data. So next, I overexpressed NR4A1 in the dorsal hippocampus of these mutant tau mice and found that control or vehicle infused animals shows long-term memory impairment, but overexpression of NR4A1 reversed long-term memory deficits. And this is consistent with the CBP activation work that we did with the other mutant tau model. Now, my previous work also identified these ER chaperones as direct targets of the NR4A pathway. And I also found downregulation of HSPA5 and PDIS6 after learning in the tau mice. So next, I investigated whether expression or overexpression of HSPA5 is sufficient for memory rescue. Now, I had to control the expression of HSPA5 because over too much expression of HSPA5 might be deleterious to the neurons. So I found that overexpression of HSPA5 in the tau mice was sufficient to ameliorate long-term memory consolidation. Next slide. So to summarize this work, I found that in healthy neurons or healthy animals, neuronal activation or spatial learning leads to increased histone acetylation through CBP. CBP associates with CREB and induces its downstream gene expression. A lot of immediate early genes or activity-induced genes are expressed after learning, such as NR4A1 and NR4A2. NR4A1 and NR4A2 proteins then get recruited on their target gene promoters and regulates expression of genes such as HSPA5, PDIS6, and several other genes encoding ER chaperones. These ER chaperones then resides in the ear and then form a multi-protein ER chaperone complex, which help fold newly synthesized polypeptides such as GLUN2A. Correctly folding of this protein helps it to traffic onto dendritic surface or postsynaptic density compartments and facilitate long-term memory consolidation. But in Alzheimer's disease and related dementia neurons, we found that learning-induced expression of or learning-induced histone acetylation was impaired, mostly because of reduced CBP expression, which led to downregulation of NR4A1, NR4A2, and several other immediate early genes, but also downregulation of NR4A target genes like HSPF5, which led to reduced protein folding and ultimately long-term memory impairment. So this work identifies a novel machinery of protein folding, which was previously underappreciated, and this study also connects from the nuclei where chromatin changes, like epigenetic changes on the chromatin, lead to expression of genes, and then we went out of the nuclei to the chaperones, and then protein folding, and then how those proteins are trafficked onto the dendritic surface. So lastly, I'd like to acknowledge Dr. Ted Abel, who is my mentor, and he provided me like tremendous support and like encouragement, and gave me all the independence to explore, investigate like the different aspects of my research. Utsa Mukherjee helped me with the cultured neuron experiments. I'd like to thank all my collaborators, Jake Michelson and Ethan Ball. They helped us with the bioinformatic analysis from the RNA sequencing results, all the fantastic core facilities, and my funding agencies. NIH, National Institute of Aging, funded my K99 R00 grant, and some of this work was also supported by the Nelly Ball Trust. And thank you very much for listening, and I'll be happy to take questions.
Thank you so much for presenting your amazing work. This is such, I hope everyone appreciates how much work this is and how great the methods are that you used, and you just didn't stop because of one method. You needed to switch to another method, right? You just used all the methods that you needed to answer the question.
Right.
So yeah, it's amazing. And I'm glad you had this opportunity and this grant to help us solve these questions. So
Oh, absolutely. Yeah.
it's amazing. Um, so I wanted to ask you, um, just I saw that NR4A seems to be. I found a paper that it seems to be also involved in addictive type of behavior, like cocaine-induced
Mm-hmm.
behavior. So do you assume that this is a very general mechanism in excitatory neurons, also inhibitory neurons, in all kinds of brain regions? Because your work was focused on specific brain regions, but do you think this mechanism is probably a pretty general one that you could probably target for different type of disorders, right?
Yeah, that's a great question, and honestly, we don't know the answer. Uh, I mean, because I have investigated only in the hippocampus, and after only a particular task, which are regulated or which are dependent on the hippocampus. Now, outside hippocampus, NR4A expresses in dopaminergic neurons, and it is also involved in other biological functions like stress and DNA damage response. But the ChIP-seq data that I showed, where we found that NR4A1 is enriched on the promoters of HSPA5 and PDIA6, they were obtained from T cells. So I would believe that this would be a more general mechanism by which, in addition to all the different other functions that NR4A might have, this regulation of protein folding or genes encoding protein folding would be a more general function of NR4A.
Yeah, that's really interesting. Do you think that one day, if it's a general mechanism, that one day so do you think we could detect earlier on in life a vulnerability towards having basically different levels of NR4A1, and by that basically detect early on a vulnerability to addiction, Alzheimer's, and so on that we could address? Let's say, in the future we can have RNA sequencing very cheap for $5. This will never happen, but
No, I completely agree with you. I completely agree, and that's actually currently what I'm doing. And Jamie was asking me if I mean, if I can come back later, and I'll be happy to come back later to talk about like the more recent work that I'm doing. This is something I'm chasing currently, where I'm investigating the different population of cells in different regions of the brain that shows induced expression of immediate early genes, and I'm focusing on the NR4A genes after learning. But it will also help us to understand the cooperativity of NR4A with other transcription factors and other proteins where they are co-expressed and co-regulated, and maybe also associated with other functions. And like with all the new techniques, like of course the single cell sequencing, multiplexing, Murphy spatial transcriptomics, we have like a lot of resources these days to chase this question, to address these questions in a very precise manner. But again, I mean, all my experiments I'm performing after spatial learning, and I'll be like happy to collaborate with someone who can like who has experience with addiction and other behavioral aspects.
Yeah, I would just imagine that maybe, also PTSD, that it points towards maybe a population that has a lot of flexibility at some point in life, through stress or something that happened, that the flexibility basically of learning new environmental, the
Yeah,
environment
yeah,
is
yeah.
safe now.
Hmm.
Yeah, yeah,
Yes,
would be really.
absolutely. Absolutely. It, I mean, NR4 expression changes after various different stimuli or different conditions also, like epilepsy, stroke. Uh, in autism, it has some connections in autism also. Not that we understand it very well, but it's worth looking into it.
Yeah, maybe the Department of Defense would be interested to have a screening of PTSD, for example, or
Right.
maybe depression, and because it kind of, yeah, I kind of can imagine that this could be a good indicator actually. But I'm pointing towards the future and taking the stage. So please, everyone, flash their mics. Yeah, Serena, go ahead.
Wonderful talk and very fascinating, and such a vivid view of the details of the actual process of the transcription cascade, and very interesting. I had two questions, an upstream and a downstream one.
Yeah.
In terms of the upstream, so as far as the activation of CBP, is that derived from just the activity itself, or are there additional factors? And then I'm thinking of astrocytes in terms of at the synapse level. In some cases, with long-term potentiation, there's additional factors that lead to upregulation of AMPA and NMDA and so forth. But are there, as far as the pathway of CBP activation, what's it look like upstream from there?
I mean, CBP binds to CREB, which is cyclic AMP response element, which is activated by cyclic AMP, and it is activated by MAP kinase pathway. So neuronal stimulation does induces expression of, or sorry, activation of this CREB CBP through cyclic AMP dependent processes, through MAP kinase dependent processes. Now, how CBP autoacetylation induces after learning is not clearly known, but we believe that binding of CBP with CREB uh, or I mean, like its CBP confirmation changes happens, which makes it like more feasible for it to like acetylate its target genes. Uh, but yes, it's upstream regulators are cyclic AMP and kinase pathways, and it is induced by neuronal depolarization and also neurotrophins like BDNF.
Interesting. And it was, uh, this happens about an hour after the learning episode. Is in conditions of where there's continual learning or so, or there isn't really a break. Does it? Is there some type of
So the
A
time
steady
point
state.
after,
Mm-hmm.
yeah, the time point after learning is again it this gene expression follows like a wave-like pattern, but the initial activation is phosphorylation of CREB. So CREB phosphorylation occurs within 15 minutes after learning, like 10-15 minutes after learning. There is a hyperphosphorylation or of CREB, and then CBP gets associated with CREB, and then we start seeing gene expression changes or this induction of or the beginning of the wave of transcriptional events within like 30 minutes after learning, and then it
Mm-hmm.
reaches its peak between 30 minutes to 1 hour, and then starts going down from 90 minutes, and within like 3 hours time point you see that the initial wave is down, and then the second wave appears after 6 hours after learning, which are basically the target of the initial like wave, like ARC and several other immediate early genes, which are induced in the regulate expression of the second wave genes.
So does it follow then that for optimal learning, there is some timing in terms of presenting the material to be learned, and allowing this process to happen before you're loading that up again with different things to learn? Or does
We
that not follow?
You mean two different learning paradigms?
Well, if does it make sense to for optimal learning, to optimize the process, presenting the experience to be learned, but
Mm-hmm.
then allowing a downtime for this expression cascade to consolidate?
Absolutely, absolutely. That's exactly what we do. We train the animals and then put it back in its home cage, and we make sure that the animal like doesn't have any distraction. We keep the room very quiet, and like so that it has like the best optimal environment or everything for like consolidation to happen. Because if there is another somebody entering the room, it's again a different kind of learning, so it might interfere with the previous learning event. And to enable these genes or consolidation process to happen like properly, uh, it's better that it doesn't learn a new thing or new experience after the learning sessions. Does that answer your question?
Wow,
It
that's so
does.
weird. That
It
that is
does.
like a
Thanks.
strict, rigorous control of memory in a very organic way. It seems, it just not to sound too crazy, but it reminds me of the '60s experiments on humans where they tried to control for a lot of parameters. Although there were many flaws, it still makes me think of this very like very clean and I guess like reproducible. In terms of the variability of the organisms themselves, I'm not sure if you addressed this in the talk, but what was the genetic variation, if that was something that you kept track of?
By genetic variation, you mean like SNPs, mutation on the
Yeah,
genome?
yeah.
I No, we mostly focused on the NR4A transcription factors, which we found to be regulated by Krebs CBP pathway. And there we made some mutations in the NR4A transcription factor or transcription activation domain. But otherwise, we didn't make any changes on the genome. We were like most interested in understanding the transcriptional waves, and then what is the precise signature of gene expression after learning, and what are the mechanisms or regulatory mechanisms that controls these precise events after a learning experience so that memory consolidation occurs properly.
Yeah, no, I'm just asking because there's this one paper that I find somewhat annoying, irritating sometimes when I think of the complexity of genetics. There's a
Mm-hmm.
paper by Manolis Kellis that famously talks about how obesity can be accounted for by one mutation, one letter out of uh, billions,
Mm-hmm.
and that can account for obesity in some individuals to a large extent, beyond even their control. And so I always think of the implications of how much variation there is, or to what extent we're keeping track of that variation with organisms. But
I
that
mean,
was kind of the motivation of the question.
Yeah, it could
That's
be possible because if the mutation is present in one of the active domain of a protein which plays important role in a certain biological function, that mutation might change its whole function because it might interfere with its ability to perform its function, and that might block. I mean, I can think of like CREB. If you activate CREB by certain mutations, you can lead to it. It might lead to memory impairment and synaptic plasticity impairment. This one mutation that I'm talking about in the tau model, the P301S or P301L mutation. Now, this is a single point mutation which is seen in patients with frontotemporal dementia. Sorry, I forgot about this one. So here in this mutant tau, it gets hyperphosphorylated, and that hyperphosphorylation of tau leads to neurofibrillary tangle formation, and our these animals shows all the like most of the pathological signatures of Alzheimer's disease, including memory impairment. So it is possible if the protein is a important component and if the mutation can affect its function, for example here in the tau mice, it could lead to deleterious effect.
I was gonna ask real
Chris?
quick.
Oh, sorry.
Yeah.
I saw in your studies you were focused on memory consolidation. I was wondering if you looked at extinction at all. The reason I ask is there was a paper I noticed. It came out a couple months ago, but I didn't pay much attention to it until I heard your talk. That showed work with in the infralimbic prefrontal cortex, where they
Mm-hmm.
showed long non-coding RNA regulating extinction memory, and specifically NR4A2. So that might be an indication that that there's some
This
work.
is from Tim Reddy's lab, Australia.
Yes. Yep.
Yeah, yeah,
Yep.
right. Yeah, Tim works on like extinction and NR4A2. Yeah, that's a great question. And the thing is that in our mouse model, we don't see any memory consolidation. So we basically cannot do much of extinction work. We try to do some with other proteins involved in transcription activation, but with NR4A or CBP mutant mice, they show memory impairment. So we couldn't investigate the extinction. I know that in addition to NR4A, there are like HDAC inhibitors, which are HDACs, histone deacetylases or lysine deacetylases. They are also essential for extinction to happen normally. And yeah, Tim is doing like fantastic work with long non-coding RNAs and like regulation of NR4A in extinction related behavior.
Awesome. Thank you.
Yeah, thank you so much. Such a fascinating work, and to me, it was very interesting in the case of the NR4A nuclear receptors, because I know that we have expression of them as in a T and B lymphocytes as a gatekeepers of the immune tolerances. And you just mentioned about the Alzheimer patient, but as long as there is a relationship between the some of the patients who are struggling with the AIDS and HIV, and there is a CCR5 related with this region. So that was very interesting to me, because still in a clinical level we can find some relationship between the immune compromised people
Mm-hmm.
and those receptors. So thank you so much for sharing your work with us.
Yeah, thank you everyone. I mean, all the questions are like really interesting and they're very helpful.
Hello, Doctor.
Hi.
Can I just say that that was an absolutely incredible talk. It's quite exciting what you're
Thank
discovering
you.
here.
Thank you.
Um, and one of the questions I've got, please forgive me if it's a simple one, but when you were discussing here, like reverse long-term memory deficit and things like this, one of the things
Mm-hmm.
that I started to wonder, um, that's incredible. Okay, reversing memories like this is incredible. Is this indicating that a great deal of things like memories are actually stored, still stored? It's only the bridge between the storage and retrieval that's the problem then, as opposed to something that just was never remembered properly, if that makes sense.
That's a fantastic question. That's a really good question. Uh, so the answer to this question is we are not bringing back a memory which was lost. Okay, so these are like memories that are formed after we did our manipulations, like drug treatment or virus expression, so that the memories can form properly. In mutant mice model, this NR4A model, or in Alzheimer's, a new experience following a new experience, memories cannot form because of several different reasons, and one of the important reason is transcription deficits. There is impairment in transcription after learning, which makes it like unable to process those short-term or sensory information to make it more stabilized and form long-term memory. And that's one of the problems associated in Alzheimer's with memory consolidation. Here what we are trying to do is we are trying to enhance those machineries, or activate those machineries, so that after the learning event memories can form naturally. But the animal, if whatever it or if a subject learned something before and then that memory went away or degraded, we haven't tested our strategy in that model. So I mean, that's a great question, but so far what we are doing is only the new memories that are formed, we are trying to make or stabilize the new memories in Alzheimer's disease, and also in
That
aging. Yeah.
That's absolutely fascinating, and I'm wondering, you maybe haven't explored this yet, but you know how there's so much material out there, some of it not very useful, but on things like how to memorize things better, how to learn faster, things like that. Have you looked at or found anything that would indicate like in the future you might have a refined process for humans to optimally learn, and then maybe with a supplement maximize retrieval, or so? Because it sounds to me that this is going to be a mixture of um like say like me reading a book, right, and then the proper behavior, and maybe even diet to maximize the recording of the memory, but
Mm-hmm.
then some kind of supplement to strengthen the connection, the retrieval process. Do you think that that's something that you'll be seeing in the future?
Yeah, again, a really good question. So, what I haven't talked today is about sleep. And one of the most important component of memory consolidation is sleep, because that's actually what we believe is during sleep, our memories are are stabilized. And in TED's lab, where I currently work, they have seen that after the spatial learning protocol, if you sleep deprive those animals, they fail to acquire memory. They couldn't consolidate memories. And in Alzheimer's disease patients, it has been reported, like several from several different groups, that sleep is actually affected. People with Alzheimer's, they have fragmented sleep, disturbed sleep. And if you can sleep for like 8 hours, you will have good memories, you will have a strong memory. So sleep is, I mean, diet, yes, perhaps probably, but most important is sleep, because sleep plays active role in memory consolidation.
That's fascinating, and we're still coming, like we just discussed, multiple speakers that have came before you have all brought up sleep as being important, sleep, 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 and
Yep.
how crucial it's actually in our development. So it's incredible that you're offering another um, piece of that jigsaw to that. So thank you very much for your time, and thank you for letting me ask questions.
Yeah, thank you very much.
Oh, hi, Jody. Yeah, thanks for your wonderful presentation. And this research, I also in a second, Karina. This is the all so many stages of logical links, and you. I mean, congratulations just for persisting through. I learned a lot, but as a layperson, I do have a few questions. If you allow me that for the experiment,
Mm-hmm.
I just want to understand a little bit better for the retrieval stage that you what are the yellow circle that entails in detail that is it so I'm thinking so that your learning is for spatial right memory, then it must be a format of maze that of different paths or something, some novel objects for the mice to play with, or
Yeah, I mean it's a very simple like strategy. So we have an open field, and there will be objects at specific spatial location, and there is a small queue inside the open field. So the colored circles were objects. So there are like three different objects. They are made of glass so that there is no smell or anything, and the animals will explore the objects to encode the information of the spatial location. Now what we do is we move during retrieval, we move one of the objects to a novel spatial location, and if it learned the location of those objects, it will, I mean, rodents are like they explore anything they find novel in their environment. So if they understand or if they remember the location of those objects, they will recognize that one of the object has been moved to a novel spatial location. So that would be like a novelty to them. So it will explore the moved object more. Same thing you can do with like novel object recognition, where you replace one of the objects with a new object and check whether it explores the new object. The spatial object recognition is more dependent on hippocampus, whereas the novel object recognition is not fully dependent on hippocampus. That's why we used the spatial object recognition task. I mean, it's a very simple behavior, but Morris water maze is more of a maze, where it's spatial navigation memory, where there will be a platform, a hidden platform, and animal have to like explore using spatial cues in the room. It has to find the hidden platform, so it takes more time to train the animals, like 3 to 5 days of training. Here it's just 1 day of training, and like less stress, because we were careful, we were tried to like control for any sort of stress to these animals.
And I just want to say, fuck yeah, for controlling for stress for the animals, because that is awesome. I love to hear that, because it's difficult. I think doing this work, I think the moral implications oftentimes are overlooked by this necessary work. And so I
Absolutely.
really appreciate hearing that. And apologies for the language, but just it was awesome.
yeah, okay, thanks. So the just again for the so the spatial information encoding is not with the object itself, but the trajectory or the path of the maze, the mouse. Is it, or is it because of the didn't encode? For example, in the case of memory loss, that means the mouse forget the. Uh, I mean, it found the same object novel again. It's. I just wanted to understand what is being encoded.
So it's a spatial memory. Suppose we go to a novel, I mean, a new place. Okay, we are driving. I mean, forget about like GPRS in olden days. Okay, we are driving across a street, and we like see a big tree on our right. We take right turn from there, and the destination is on the left side. So we encode those spatial informations in our hippocampus. The next time when we go there, when we take when we see that tree on the right side, it will activate those neurons. Okay, and it will activate, and then we know, okay, from here we took the right turn, and then our destination was on the left side. So that's the hippocampus encoding memory. So that's the retrieval which we are doing. We try to recapitulate the same thing in animal model with making it a more simplified like task. So it's all about like location. Hippocampus constructs the location as a map in its system, and whenever the animal or an individual passes through the queue, there are activation of neurons. Of course, it starts with entorhinal cortex, where it's filled with grid cells, and then signals to hippocampus through like perforant pathway, and there are like trajections. But the hippocampus actually encode the spatial information. and that's the information, or that's the memory which we try to like assess.
Great, thank you. That's very clear. Thank you. So on the slides number 9, that you show the nuclear NR and receptors, the among the candidates. So
Yeah,
you have actually you mentioned that 14 of them, 13 of them are actually show response in the first 2 hours of training.
yeah.
Then I wonder the rationale behind, for the 1 hour selection of the NR4A for 1 hour, and
Uh,
the timing is specific.
I mean, the right side experiment was done later, so this work we try to like investigate all the nuclear receptor genes that are induced after learning, and we found these NR4A genes to be induced within like 2 hours after training, but the expression goes up within like 30 minutes, and at 1 hour we see highest expression. Now, why I am interested in NR4A is because of my previous work with CBP, where where I like consistently found that these NR4A proteins are regulated by CBP. NR4A promoter has CRE elements, which are like regions where CREB protein binds, so they are direct targets of CREB CBP pathway, and that NR4A genes are induced after various stimuli, and several groups have showed that targeting NR4A if alters behavior. So those are the reasons why I pursued with NR4A, or I investigated NR4A, not other nuclear receptors. I mean, they are also equally important or is interesting. I'm sure there will be like the other 13 genes which are induced after learning. Some have actually been investigated for their role in memory, and some have been investigated in Alzheimer's, especially with amyloid beta models. But and in my tau mice also, I do find some of these nuclear receptors to be downregulated. But the reason for NR4A is it is a potential target for memory enhancement. It is regulated by Krebs CBP pathway, so it is epigenetically regulated. And as it is epigenetic regulated and it is a target for memory, there are a lot of different scope for therapeutic approaches or therapeutic interventions. We can use epigenetic drugs, we can use CRISPR mediated gene therapy. There are like a lot of different options which we can investigate, and focusing on this particular subfamily will help us to understand, like in a broad way, in a broad platform, how activity induced gene expressions are regulated during memory consolidation, and how it can go awry during like different diseases associated with memory impairment, so that we can come up with a mechanistic understanding of what goes wrong, and how those components participates in memory consolidation or important biological functions.
Understand? Yeah, thank you.
Mhm.
I had a really one last quick question. I was thinking about this because of what you said about the sleep doctor, and this might be a little bit out of scope, but I wouldn't allow myself to not ask. You mentioned before how important sleep is to this process, right? And you mentioned like 8 hours sleep. I wondered, do you think when in learning something, any benefits could got from learning a thing and then having something like a power nap right after it? Or were we talking about it needs to be like a deep, long-term nightly sleep or something like that? Does that make sense?
Yeah, I mean, it's more complicated than we think. Okay, so and not also it's fully understood. Like in sleep, there are like two different like phases in sleep, which like constantly shuffles REM sleep and non-REM sleep, and there are like this. I think in REM sleep. So I mean, they have their different functions in sleep. After a training, if you like sleep or take a power nap, that might help, and that might be a good idea, but I cannot support it. uh, it could. What is important is focus. Like after you learn something, even if you don't sleep, if you try something else, your memory will be diluted, because you are learning a different task, so you are not giving sufficient time for the consolidation of memory from the previous task. It could be true for like a lot of different behavior that we perform every day. I mean, I talked about sleep in a more broad way. I don't know whether these like transcriptional events after learning require sleep. It probably doesn't, but it probably sleep acts like in a more global way to like function as a stabilization for neurons, to control the damages. There is a study from Liwei Sai where she found that learning or new neuronal activity leads to DNA damage. Okay, and DNA damage opens up the chromatin and facilitates expression of immediately genes, and then they are repaired back. So in aging or there is an accumulation which is a age associated memory decline. There are more accumulation of damaged genomic regions, and what a lot of people believe is that, or what is true is that during sleep our brain goes to a more repair function. So our brain is more active while we are sleeping because it functions, it participates in repair machineries. So it doesn't directly correlate with memory, but as neuronal function, it is more important because it helps the neurons to be like more polished, to be more active, or more in a better state to be activated further for or ready for new memories to form. I mean, if you go into like where memories are stored, the discussion will never end, because if you just imagine about like one neuron, or like where we are learning something, okay? So genes are induced, we are learning something else in one day. How many different things we learn, and where are all those memories stored? We have no clue. So we just started to understand this machineries and mechanisms of how memories are stored and where they are stored. And like sleep probably functions as a damage control device, and also stabilizes neurons for like activation and function in next day. Did that answer anything, or I just talked crazy?
Oh no, no, it did. That was incredibly interesting and very exciting.
I mean, I have a
This is so
problem of
so good.
talking like into like more hypothesis based things and philosophical stuff, so I can get derailed like very soon, very fast, and
No, no,
talk about a lot of philosophy.
no, I love it, and I think we all love it here as well. Thank you. No, that's a great answer. Thank you so much. Thank you.
I wanted to ask one quick question. That's all right.
Sure, please.
I know Serena said asked something earlier about what is down and upstream here, so hopefully I'm not repeating any of her questions. But I know the Krebs activity can be modulated by many different pathways, um, G protein pathways and whatnot. I'm curious if there's any, upstream of all this, this pathway that you're exploring here. Is there any, is it known how coupled it is with electrical signaling? Like, if is this something that is directly driven by local potential, or is it more of a like a downstream chemical pathway?
Any that again, good question. Any form of any stimulation that would induce neuronal activity. It can be long-term potentiation, it can be depolarization, it could be like electric stimulation. Any any component or any stimuli that would lead to neuronal activation in terms of calcium influx or BDNF or cyclic AMP activation would lead to activation of Krebs. So Krebs is like a signature or mark for neuronal activity. So yes, long-term potentiation or neural depolarization does induces Krebs activity, Krebs phosphorylation, or even electrical stimulation should also induce.
Awesome. And the last thought I had was with this particular pathway, is there anything known about subcellularly what's happening? For example, is this been shown in all synapses, like very distal synapses in pyramidal neurons versus maybe somatic or synapses that are nearby? I'm asking because I know there's a lot of machinery all the way out in distal dendrites for transcription. So I'm wondering if this is something that's happening everywhere in the cell or if it's concentrated near the soma.
You mean the which machinery? Activation of CREB happens in the nucleus, whereas the gluon to a protein distribution happens in the synapses. So for all the transcriptomic experiments, I focused on the nucleus, and their activation of CREB, CBP happens. Like local translation happens. For example, ARC mRNA is translated on the dendrites, and there it participates in synaptic plasticity. But I haven't focused on the synapses much, except the like protein folding machineries, and that too I just focused on the postsynaptic density compartment in cultured neurons, where I just simply investigated gluon2A NMDA receptor surface distribution. Otherwise, I haven't looked much. The only closest to the synapses would be the LTP experiments that we did with the phenyl butyrate or chemical chaperone, where we stimulated the Schaefer collateral pathway and then investigated whether the chemical chaperone rescues long-term LTP. But that's really not what you were asking, right?
Yeah, so I guess it's not really known whether the CREB can happen in the dendrites. So the assumption is that it's being trafficked from the soma.
Right. Yeah.
Yeah.
A crab, like crab in the dendrites. I don't. I think
CREB is a
that's
nuclear
the
protein, right?
nucleus. Yeah, it's
Yeah.
only in the nucleus.
Excellent. Cool.
Just to follow on to that, I was also interested in the downstream. So the consequences are upregulation of glutamate receptor, but is there any? And it's a similar question to what Wisdom was getting at, but um, is there any spatial specificity to where those receptors end up, or is it sort of a non-specific? Or are they more directly trafficked to the synapses of? I mean, is there a retrograde loop there that the synapses that cause the stimulation are going to get more receptors?
Uh, Great question, Serena. We don't know whether the glutamate receptor expression is regulated by this pathway. What we know is the protein or the folding of NMD receptor only glue into it. That's what I have investigated. I haven't looked into the other targets of this ER chaperone pathway or proteins. So we know that the gluon2A or newly synthesized gluon2A protein folding is facilitated by this NR4A downstream targets, and I mean again only in cultured neurons which were depolarized with KCl, we found that there is a like increased postsynaptic density compartment colocalization of gluon2A. It seemed to be like a broad, non-specific activation, but we haven't investigated it further. There is like a couple of experiments that we did, and these are like more recent findings. So there is definitely scope to investigate it further. We are currently doing some STED microscopy to look into like specific synapses and how specific this gluon2A export or trafficking is, and there is no single cell protein, or I mean I haven't seen any single cell protein like experiments that we could do. I mean, if we can do some of those experiments at single cell level at from a given brain slice or something, it would be interesting. We just started to like investigate those questions. So I mean, sorry, I don't have much answer
Oh,
now.
well, thank you. Yeah, it may just not be known, but it's kind of curious about how much, whether the the source of the amplification, and and Wiz mentioned, is it by the soma or out on the distal dendrites, but
Mhm.
how much credit assignment is happening there specifically, and is it spatially localized?
Right, and most of those answers are not known, and there must be very strong like regulation and precise regulation for like different subregions.
Well, maybe there would be an option to do two photon microscopy with electrophysiology and do line scans, and then have specific blocking, maybe with uncaging, combined with uncaging, and then while you uncage, to have specific antagonists and agonists, and then perform line scans in the distal versus uh, more proximal um, dendrites, and see if you can detect like differences in signaling, maybe maybe like very sensitive calcium dyes that you could fill the cell with, and then do some line scans. But I'm not sure if they are. You would probably need to design a caged drug for those specific receptors. I'm not sure if there are any out there.
Yeah, that's a great suggestion, and we did thought about like two photon imaging, but for caging, what approach you are talking about?
So uncaging means you have the drug caged. So you in the lab when I was a PhD student in George Augustine's lab at Duke University, we had the postdoc that did that with compounds herself.
Mhm.
She did the cage version of drugs, and then you elicit a very precise laser. It's usually strong UV laser,
Right, right,
uh,
right.
and then
Okay.
you just
Hmm.
release the drug on that spine. So we have GABA uncage and glutamate uncaging, but then you don't necessarily know if it's that specific receptor, right? If you could do glutamate uncaging just on that spine and see differences, but maybe you could have. Yeah, I would need to think a little bit about it. Or you just ask for a chemist to make your caged version of a
We
agonist.
actually have developed a like caged version of this PDIS6 because that's another target we are chasing, and using UV. But I mean the goal is slightly different there. Like with UV radiation, it will be caged, so we could capture all the proteins with which it interacts. Uh, it would be
Oh,
it's an interesting idea to investigate the glutamatergic neurons or the synapses, and then distal and all those questions. I mean, this is great, and this is great feedback,
yeah,
and
because it's just
everybody
very
thank.
hard to do patch clamp on distal dendrites. It's very, very hard. And uh, so usually what you prefer to do then is line scans, but then you have to somehow be also precise in the release of some activator or inhibitor of the target you wanna
And
you
there are
wanna
not too
miss.
many like specific molecules for this proteins. There can always be off target effect. That's what I'm always like worried about.
Ah, okay. Yeah, yeah.
But yeah, this is great, and I mean I would have to collaborate with an electrophysiologist to do some patch clamp or two photon imaging.
Yeah, I wish I don't have I'm not in that lab anymore where I did that. I used to do patch clamp, went to photon for a long time, but I don't have it anymore. But
So
yeah,
Kathleen,
there are
I wanted
many
to
more.
I wanted to mention something. So I'm in the job market and I'm in like currently negotiating, or I just finished my first final round of interviews for the assistant professor position, and I'm expecting an offer soon. So I mean, if you know anyone who is interested to, I mean, I'm saying everyone here. If anyone is interested to like investigate these things that I'm working on, memory mechanisms, Alzheimer's, transcription, epigenetics, uh, I will probably need people. I'll be hiring. So I mean, this is not official, but just wanted to mention.
The
Just
unofficial official announcement. Love it.
I'm sorry.
It's unofficially official, but it's just I'm just kidding.
I I'll get to know next month. The committee hasn't decided yet. They haven't met yet. I have a good feeling. It went well. I have a good feeling about it, and I'm expecting a positive answer soon. But I'll like to prepare like before, like I mean as soon as possible, so that I'm well prepared when I start my own lab.
Yeah.
And the most important factor is hiring good people who are interested and motivated, and yeah, that's why.
First of all, congratulations.
Thank
They
you, but
would be
not
silly
yet.
if they wouldn't take you. So I'm very upset. I mean,
Thank you.
and yeah, if you have a job post or some job description, I can add it to my bio. I can, we can add it, and then I also have, I'm an alumni of a for international PhD program where we share a job post, so I can send an email out to share. And those are very, very good, precise selected students from Europe. So
Yeah, that would be great.
that,
That would be
yeah,
great. Yeah.
so yeah, send
Thank
me
you.
and then I'll be happy to share it. I cannot promise anything,
Of
but
course. Yeah.
I'll be happy to share.
No, thank you. Thank you very much. I will let you know.
if you get more serious about the synaptic dendritic stuff, let me know. We can talk offline or something. I study calcium signaling and dendrites, and I'm always thinking about what's downstream of my measurements. So yeah.
Sure. Yeah. I'll write down your name. Thank you. I mean, we do have all those equipments here. We have at INI, we have 2 photon, we have patch clamp, like everything, a lot of central facilities. So yeah.
That all sounds incredibly exciting, and hopefully, anybody who will be able to listen to this replays might be themselves someone or know someone who would want to be part of this. I know if I had the kind of expertise, I would be absolutely racing towards you to do that. It just sounds
That's so
so
kind.
exciting work.
That's so nice. Thank you, Jimmy.
Yeah, and please come. Yeah, we can also chat offline when you want to come back to present exciting updates, and then we
Yeah,
can
of course.
meet
Yes,
again.
I'll let you know. There's something I'm cooking now. I mean, very interesting ideas, but yeah, once my paper is submitted, I'll probably send you an email or contact you here to like set up another time, another date for a presentation. Some interesting things that I'm working on, which I'm sure you all will be interested in.
Yeah, that's wonderful. I'm really looking forward to it, and I love to hear that memory, epigenetic mechanisms, and all this work is making such advancements because yeah, I was very interested when I was still in a lab with 2 photon and epigenetic mechanisms and memory, and maybe using different, very specific targeted cancer drugs that
Oh,
target all these epigenetic mechanisms and try them out if they would actually help in different disorders. So yeah, I find it
Yeah,
very
that's how it started, right? Like cancer
exciting.
treatment, HDAC inhibitors, epigenetics, and then slowly it shifted to neurodegeneration and memory mechanisms. So
Yeah, exactly. Yeah, yeah, I was interested in that for a while. So yeah, it's very exciting to hear. And yeah, I didn't get the grant on that, but I'm really glad that that worked out, and it's really exciting to hear these new breakthroughs in neuroscience that
mhm.
will most likely help people in the future. So it's really wonderful. So thank you for coming and sharing your work, and I wish you all the best for your new lab, and I wish you the best of students and postdocs and all the grants and the least bureaucracy to start
And
things.
all the funding
All
you
the
could ever
fun.
want.
All the fun things, the least bureaucracy,
That's the most important thing. Yeah.
and
Yes.
your mice breathe well.
Yeah. Oh my God. Yes. My spirit bed. You
That
know
you
the
don't have
pain.
to, yeah, that you don't have to rebuy them.
Yeah. Yeah.
Yeah.
But
Okay.
thank you so much. I mean, really, I had a great time here. It's a really good, it's a great group, and all the questions and discussions, it's really nice, and I took notes and very helpful, and I love to come back here again for another talk. But I'll also participate in the other talks in the upcoming weeks, and be a part of this science society. So thank you very much for having me here today.
Thank
We
you.
will
You're
absolutely.
already
Yeah.
the best moderator because you led me to announcing the next room. If that's okay. That was the perfect transition. So yeah, thank you everyone for coming and asking great questions and contributing to this discussion today. We really appreciate that, and we will have actually Dr. Garin coming tomorrow and talking about the human brain evolution of abstract thought. And I know Serena will ask something about the
Oh, yes, more astrocyte questions, certainly.
and then we'll have on Wednesday Dr. Motri talking about complex oscillatory waves emerged in brain organoids. And it's a lot of neuroscience this week. It just came that way. And Dr. MD PhD Zuniga talking about Alzheimer tautopathies dependent on RNA surveillance. So it's really close. Well, you know, fairly related to what we heard today from you. And then Dr. Henry, he will talk about the thermovoltaic that he developed that has a efficiency of 40%, which is quite high. So yeah, thank you so much, everyone. A special thanks to you, and come back.
Of course, I will. Thank you.
Okay, bye,
Thank you.
everyone.
Bye bye.
I'll close
Thank you
the
so
room.
Thank you.
much, Doctor. Be well.
Thank you.
Thanks, everyone. Thanks,
Thank you.
Doctor.
Thank you. Bye bye.
Night, everyone.
3, 2, 1, bye.